To run IGV-ScanFold, it must first be downloaded from GitHub using the link in the paper. Once downloaded, it will appear as a zipped file, and that zip file should be moved to your directory of choice. Once moved, that file can be unzipped, and it will display either an IGV-ScanFold icon on Mac or an IGV-ScanFold folder containing a RunMe file on Windows. To launch the program, you will double-click on either the icon or the RunMe file, and it will begin launching the program. It will then take you to a new window where IGV itself will be launched. This process can take differing amounts of time depending on the computer being used, but once it has launched, it will begin downloading a genome from the IGV server and then loading that genome into the IGV window. Once that genome has loaded, we can then begin the process of running ScanFold. The first thing we want to do though is check that we are using the correct genome, and to do this we will go up to the upper left hand corner and check that we are in the correct genome. So currently we are in the human HG38 genome, but if you want to look at genes in some other genome, you can simply go up to the Genomes tab and either load a genome from a file, a URL, or from a server. Now we'll load it from the server, and here you see all of the genomes that are available from IGV, and for our purposes we will be using the human HG38, so we'll select that and click OK. Now if you want to run a genome that is from a file, this will be uploaded as a FASTA file, and that FASTA file will contain your sequence of interest, and once it's loaded, once again we can perform our ScanFold runs in the same way as you would for a gene in the server. So to select your gene, you'll go up to the search bar, and you can either enter that gene name or the chromosome number and coordinates. So for our purposes, I will type in MIC. This brings up the gene cartoon. I'll go ahead and drag the cartoon up to the top here and zoom in on the gene itself. So once you're zoomed in, you can then do a scan on the entire visible region by clicking the ScanFold tab, followed by Run ScanFold on visible region. And this pops up a parameters window for ScanFold. All of these parameters are set up to give you a good quality run, so they do not need to be changed. The one thing that you will want to check is that your strand is correct. So for MIC, we are looking at the forward strand, but if you're running a gene that's on the reverse strand, you'll want to switch this over so that the correct strand is being scanned. You can also change your output folder, as the default output folder is a temporary folder, but if you want to save these to a more permanent place, you can click Browse and find the place you would like to save those, and then all of the output files will be saved there. You can also change your folding algorithm. By default, we use RNA fold, but you can also select RNA structure, and you can run multiple scans using both folding algorithms to compare the results of these. Additionally, you can change your step size and randomizations to change the resolution of your scan. So by decreasing step size and increasing randomizations, that will increase your resolution, but that will also increase the time that it takes for the scan to run. So for the purposes of this video, I will not actually perform this scan, as it will take a decent amount of time, but I can show you how to run a more targeted scan on a select region of a gene. So to do this, we go up to the Define Region of Interest tab, select it, and then place 2 markers on the gene cartoon around your region of interest. This will highlight that region red. You will click that highlighted area and then select Run ScanFold on selected region. Now this again brings up the same parameters window. You'll want to again make sure that you're in the correct strand, select your algorithm, and either leave all the parameters as default or change those to whatever you would like. And then you can go ahead and select Run. Once you hit Run, what is happening will pop up in the log output window. So here you can see that ScanFold scan is running. It tells you the length of the sequence, the number of windows generated, and the estimated time to completion. Now this time is an estimate, so depending on your operating system and the processing power of your computer, these times may be longer or shorter. But once this is done running, all of the results will be output below your gene cartoon. So if you're doing this on the entire visible region, these metrics will be output aligned to this gene cartoon, or if you're doing it for the targeted region, they will be output and aligned to only this targeted region. Additionally, you will also be output a 2D structure as a separate entity. It'll be a pop-out window, and that 2D structure can be visible outside of IGV itself. So once all of the scans are complete and everything is output below, you can begin the process of analyzing the predicted structures.